Okay, good morning everyone. I think it's about time to start. So I'd like to formally welcome you all to the third CCP-EM Spring Symposium. Last year we were in the same place and we had slightly fewer people. In the first year we were in a smaller room with about 100 people. So I think it's clear that this meeting is getting bigger and better every year. Certainly like to thank eBIC for a great opening day yesterday, and it's great that we can do this back to back and make it a really good meeting. So we're getting bigger and more crowded. I was asked to make sure that we didn't have too many people in the lecture theatre. At the moment that doesn't seem to be a problem. Some people are obviously still digesting breakfast. But yesterday we were overcrowded and there were people on the steps, and we're not really allowed to do that. So you need to have your badge, you need to wear your badge, otherwise the bouncers will come and chuck you out. And if you do have a problem, obviously there are lots of seats at the front which you can make use of. It's not a pantomime, it's not dangerous to be down close to the stage. A couple of other announcements before we get underway. There were tours of the eBIC facility yesterday. I think a few people missed out, so there are plans to run a few more tours today. So if you go, after this session is over, if you go back to the registration desk in the Diamond Atrium and talk to Helen, she'll put you on the list and hopefully we can do some more tours today. The other thing is wireless access. There is a guest network here, but you need to sign up for that and it needs to be authorized by one of us. Unfortunately, I don't have the URL to hand at the moment. If you search on the STFC site, you may well find it. Otherwise, talk to Helen again at the break and we can get you a guest wireless access. Not that you need it because you're going to be concentrating on the talks, aren't you? So I think that's all of the housekeeping for now. So it's great to see you all here. I'm going to hand over to Peter Rosenthal, who is the chair of CCP-EM, and he's going to chair this first session. Okay, Peter?
Martyn, thanks a lot. I should say it's very easy to be chair of CCP-EM with Martyn and his team doing fantastic work. And this session is about late-breaking what's new from the organizations CCP-EM, eBIC, and EMDB. We're very fortunate to have these organizations that are helping us get our data, process our data, and archive and validate it for future generations. So the first contribution is by Colin Palmer at CCP-EM.
Thanks, Peter. So yes, I'm Colin Palmer. I work in the CCP-EM core team. So actually, rather than what's new, this is a bit of an overview of who we are at CCP-EM, why we're here, and what we do. So CCP-EM was founded in 2012, funded by an MRC partnership grant, which got renewed last year for another 5 years. And we've got a few main aims, which are to act as a forum for the EM community. We offer training and user support for various different software packages, including our own. We support application developers by distributing and disseminating their software in our suites and running training courses on their software. And we develop our own software framework. And we sum these up in this diagram as collaboration, code, and community. A few of the people who are involved on the core team, we have myself and Tom as the two full-time core team. Martyn is the head of STFC's computational biology group, so we fall in his group. Peter, as you've just seen, is our chair, Neil, the deputy chair, and all of these people on the right have been or are co-investigators on our grant. We run a lot of training workshops. This is part of our user support and developer support program. So you see we've run quite a few of these over the years now, several on Rely On. Twice now we've run this ICNIL high-resolution model building workshop. plus a few others. You'll notice most of those have been at RAL, which is here, but there's a few elsewhere, and in principle we're happy to run workshops where there's demand for them. We just need a room with some computers and somewhere with accommodation nearby. So if you've got a particular piece of software you would like a workshop on, if you want a workshop on something to happen near you, then do drop us a line and we'll see what we can do. Workshops are normally announced on the CCP-EM mailing list, which hopefully you're all already on. We organise this spring symposium, as again, hopefully you all know. As Martyn said, that's been growing every year. Last year's talks are now available on YouTube, and the proceedings from last year are coming out in a special issue of Actor D. So that's the YouTube channel. It's on the STFC's YouTube page, and you can find all the spring symposium talks from last year. I think this year's talks will end up there as well. And the proceedings from last year are coming out in a special issue of Actor D, which will be published in June this year. Tom and Paola are kindly editing that for us. And a few of the papers are already available with advanced online publication. That's those ones there. There'll be more to come in the next few weeks. We've also got some upcoming events. Next is at the University of Leeds in July. There's a workshop on exploring and enhancing complementarity of EM and molecular dynamics. It's a joint organisation between CCP-EM, CCP Biosim and the University of Leeds. There's an exciting list of speakers and that looks like it should be a really interesting workshop. I think registration for that is going to be opening within the next few days. There's also, as recently announced on the CCP-EM and CCP4 mailing lists, there's a conference on methods and applications in the frontier between MX and CryoEM. CCP-EM is a co-sponsor of that workshop. That's happening in Barcelona in September, and I think registration is already open for that one. So we also provide some computational and software support for eBIC. Many of you will have been at the eBIC day yesterday and heard all about eBIC, but it's the National EM facility located here, for those who don't know. We provide some software support, so we've helped to install and test a few pieces of software on Diamond's computer systems, including our own CCP-EM suite, Motion Core 2, a few other programs, plus Relion 2, which we installed and got working on Diamond's GPU cluster, and tried to get that working as quickly as possible. We also help out occasionally with data processing and training, and all of this is done in collaboration with Diamond's data analysis software group, and Ala is going to be giving a talk in a few minutes about some of his work on that. So on to our software package. We write the CCP-EM software suite. We have a range of collaborating developers around the country. The idea of the suite is to provide unified access to many useful CryoEM programs with a package that's easy to install, cross-platform, with a modern user interface, has project management built in, and there's close compatibility with CCP4. To just give a quick overview of what's in it. Just to point out, there's a beta release now available, which is for Linux. You can download that on our website. There's also a Mac build, which will hopefully be coming along within the next few weeks. And there are contact details. If you have any issues with it or requests, then do please get in touch. So a quick overview of what's in it. We have a top-level control GUI. This is mostly Tom Burnley's work. So there's the project manager and individual task windows. This supports pipelining for jobs with multi-stage processes, project management for the whole thing, integration of molecular graphics, so it's very easy. You can click these buttons to quickly load the relevant job-related files in Qt or CCP4MG or Chimera. And it's designed to be very flexible, so you can launch tasks in 3 different modes. You can use the full project manager, or you can launch the task windows in a standalone way, or you can run things from the command line if you want to. Some highlights of what's inside the package. We've got RefMac. This is one of many programs in CCP-EM which is adapted from CCP4. Originally written for crystallography work and is now finding very good use in the EM world. So RefMac is for high-resolution refinement of atomic models against EM electron density maps. Has a range of different other options as well. Very powerful and useful program. And we've got a nice interface for that I think Tom has mostly written. We've got MOLRAP, another crossover from the CCP4 crystallography world. This does fast docking of atomic models into high-resolution EM maps. So this is a very useful, fast way of aligning model to map, and so it's used as a stage in some of the other pipelines in CCP-EM as well. We've got DocEM. It's from Alan Roseman in Manchester, and Sony worked on the interface for that. It's for docking atomic models into EM maps at medium to low resolution via an exhaustive 6-dimensional rigid body search, and has good integration with Chimera to view the best hits. Also got a nice interface for FlexEM and RibFind. Maya, I think, will be telling us more about these programs in her talk a little later on. But this is for flexible fitting of rigid body domains into EM maps, and has a few other features which I'm sure Maya will tell us about. And Maya is also, Maya and her group are behind Tempi. This is a template and EM comparison using Python. This provides features for analyzing the fit between a model and map, among other things. And we also have Buccaneer. This is the last one I'll talk about that's a crossover from CCP4. This is a program for automated model building based on you give it a protein sequence and a high-resolution map, and it builds your protein for you. And it's already producing some very useful results in EM, and there's further improvements to come. We also have a few other tools in the CCP-EM suite. These are mostly on the command line. So we have a CCP-EM Python environment. In that we have Clipper Python. That's a Python binding around the Clipper library, which is written by Kevin Cowson and his group at the University of York. This is a useful library of general purpose routines for dealing with models and maps, and Clipper Python should be very useful. We've got Conkit, that's written by Felix and Dan at the University of Liverpool. That's for contact predictions based on evolutionary covariance analysis, which again helps a lot in the analysis of EM structures, particularly at kind of medium resolutions, I think. As we behind that, we've got a new MRC file library, which I'll talk a bit more about in just a moment. We've also got the MRC image processing system, which was developed over years at the LMB in Cambridge, and we now include that in the CCP-EM suite. Just a brief word about this new MRC file library. A few years ago, the community came together and decided that we should have an agreed standard for MRC files because they're diverged a bit, and this resulted in publishing the MRC 2014 standard in a paper. And as part of that, the CCP-EM were given the responsibility of looking after the format. We thought it might be useful to have a standalone library for this in Python so that people can access their files easily and check them against the standard. So we have this new Python library. It's very easy to use. You get access to your data as a NumPy array, so if you're used to numerical programming in Python, this is probably already familiar to you. And even if you're writing your files with other software, it's actually a very useful way to validate them against the standard. In a couple of lines, you can run this library, and it'll tell you whether your files are standard compliant or not. Beta version is out now, installed through Python's pip system, or you can get the source code on GitHub. And that's it. I'll just finish by thanking the CCP4 core team. We share an office with them, and we benefit a lot from their experience. Our collaborators across the country in Birkbeck, EBI, the LMB, Manchester and York. The MRC fund us. We have a website. You can follow us on Twitter, and we have the mailing list, which I'm sure you're all aware of. And there's a paper you can cite if you find CCP-EM useful. Thank you.
Any burning questions for Colin at the moment? Okay. Well.
So the multi-platform support, you also put Windows there. How much, I mean, is there any actual development for all these packages to be run under Windows?
So I think many of them already, certainly the CCP4 derived ones, mostly already run on Windows, as far as I'm aware in that context. So that's we can probably make work. Beyond that, I'm not actually too sure. I think the plan is that it will run on Windows eventually, but I must admit I'm not sure in the details myself.
Is there any point of spending effort in that direction? I don't think there are many people having hopes of processing their data under Windows.
Yeah, I think it's certainly not a highest priority for us at the moment, but as I said, we're open to suggestions, so if you really do want to use this software on Windows, drop us in line and we'll push it up the list.
I mean, as I say, we had the same debate in CCP4 about 10 years ago, lots of scepticism, but we find in practice that the Windows version is downloaded a lot and used a lot. And of course, people are selective. They're not going to do the heavyweight processing on a Windows laptop, but they may well want to look look through results or do visualization or just review what they've been doing. So it's quite useful to have a copy on your Windows laptop, even if you're doing your real work elsewhere. And while I've got the other mic, one thing I forgot to say is I believe we're being webcast. If all's gone to plan, we're being webcast, which firstly means don't swear. Secondly means we should use the microphones for questions so that other people can hear.
Well, that should scare off any questions, of course. Colin, thanks. So the next contribution will come from Paul Korir from EMDB.
Thank you. Actually got the pronunciation of my name right. Okay, thank you very much. My name is Paul Korir from EMDB, EBI, so, EMBL EBI, I don't know why I said that. And I'm going to give you an update on what we've been working on at EMDB and Empire, or what's new. So just a brief overview of this presentation. I'm going to start by giving some background. One of the key things we've been developing will require a bit of background. Hopefully some of you will have seen this if you've visited the EMDB hosted at EBI. And then I'm going to give a description of the volume browser, which is a main item we're developing. And I'm going to talk about file format called EMDB SFF for segmentations. I'll talk about a tool that we're developing for handling segmentations called SFFTK. And there's a backend tool called SFFTK Plus. And I'm going to talk about a web tool that we're developing for annotating segments. And then finally, I'm just going to wrap this all up and put everything together and talk about the volume browser in detail. So to begin with, if you've visited the EMDB hosted at EBI, you will see that for each entry at the top right of the page, there are quick links. And one of the things you'll see in the quick links is volume slicer. What the volume slicer allows you to do is to slice through a volume, 3D volume, and see images across the volume. Now that doesn't seem very useful, and at the moment it's not very useful. And the reason behind that is because we're using that as the foundation to build a volume browser. So what we're working on is called the MOL2CELL Project. The idea behind the MOL2CELL Project is to try and integrate data across different scales. And part of the MOL2CELL project has been developing Empire. Empire is an archive for raw images. So you could go to Empire, download one of the data sets that are available, and you could use that to try and reconstruct 3D models. And then part of that, we're also working on the volume browser tool and the segmentation-annotation tool. So the idea behind the volume browser is we want to be able to support annotated segmentations so that we can capture detailed information about volume data together with annotations that refer to the biology, so the semantics behind the data. And so this is very useful because it allows us to connect data across entries so that people can get insight into other aspects of their biology, not just the entry that you're looking at, but to look from one entry to another. And so this is just a mock-up of what the volume browser tool will look like. It's got different parts about it. I'm going to explain this in detail. So the vision is to have the volume browser. The volume browser integrates different types of visualization plus annotation information. And again, I said it spans different scales. So in this example, we have a tomogram. And this tomogram has, you can view cells on the tomogram, but there might be structures within the tomogram that are already been deposited in the EMDB, and the volume browser allows you to see the details about those entries through this entry. Now, in order for us to do this, we need to have a file format that we can capture segmentations. And that's what the EMDB SFF is. So the EMDB SFF is community-driven. And we had a meeting starting in 2012, then 2015, where we brought together different practitioners in the field and asked them, what do you think, what are the things that we need in order to accomplish this? And so the big problem we have with file formats is every application has its own file format. And the problem with that is you end up with a roadblock. Nobody's able to communicate across applications. And so in order for us to facilitate this and have free flow of data across applications, we need a file format that can capture segmentation data. So the goal is we want to have a file format that can highlight entries, you can mark parts of an entry, you can annotate the entry, you can semantically link across entries, and then you can have alternative visualizations of an entry, and then you can embed molecular information about that entry. And in order for that to happen, you need to be able to display the volume data, you need to be able to capture segmentation data, both description in terms of textual description and in terms of geometrical data. You need to be able to overlay this data on top of the image. You need to be able to present the descriptive data in a way that is useful for the user. And finally, you need to link this to other entries. And so this is what we discussed at this meeting. And so what we're trying to do is the EMDB SFF supports structured biological annotations, various geometrical representations, rendering as XML, and transforms between subtomogram averages and the actual tomograms. There's a development version. It's quite buggy. It's available at the CCP Forge at that bit link. And there's also the actual schema that's available on that link at the top. So the top link is for the schema. The bottom link is for the code. But once you have the file format, it's not very useful if you can't play around with the format, if you can't do conversions. And that's where the toolkit comes in. So there are two parts to the toolkit. There is the toolkit that's available for anyone to use and convert formats. And then we have one version which we use in the back end. Once we receive EMDB SFF files, we want to be able to then process those files and eventually render that on the browser. So that's what the SFFTK Plus is. It's got a few other dependencies which are a bit nasty. So the idea behind the SFFTK toolkit is that it allows translation for different file formats. So these are the file formats that we support at the moment: iMod, Sega, Amira, Mesh and Hypersurface, STL, MDM, and EMDB map masks. And it also has tools for annotating segmentations, and it outputs in XML, HDF5, and JSON. So you remember the EMDB SFF is XML, but the toolkit allows you to get the data in XML, HDF5, and JSON. And that's a link, again, the same link to the development version. So this is just a structure of how the package looked like. It's got different packages within it that handle the different aspects of operating on SFF files. So the notes is API for annotations. Schema is actually the API to the EMDB schema, which has been generated using GenerateDS. We've got tests and the different formats, as you see here. So there's an API for Amira, Mesh. There's an API for maps, API for iMod. The SFFTK is used in the back end, and what we do is once we receive a map file, we have to slice through it so that we can actually render the contours onto the display, and I'm going to show you that in a bit. And that we also, because we store all the images on Omero server, we then need to interface to Omero, so there's a package for interfacing to Omero. So this is a tool that we use on the command line, but then at the end of the day, we want people to be able to use this on the browser. So what we're developing as well is an annotation tool, which will be available on the browser. And so the pipeline is that if you've got your map file, you segment your map file, you could upload the segmentation directly if it's a file format that is supported, and then it will be automatically converted in the background. Or if you have the toolkit, you download the toolkit, you install it, and you run it through the commands, you're able to generate the SFF file. You could annotate it on the command line, or you could annotate it on the Segmentation-annotation Tool, which I'll show you mockups of in the next few slides. So after you annotate it, you can then download the SFF file, and then you'll include that with the deposition or you include that with deposition of raw image data into Empire. And then it'll be processed and eventually be rendered onto the volume slicer. So if you remember, the volume slicer was that fast, sort of not very useful way of slicing through an image, a 3D map. So this is how the command line tool looks like. We've got a bunch of different sub-commands. So we can use the toolkit to search the EBI's ontology lookup service to find standard terms that you can include into the segmentation. So then you can list, you can show, you can edit and add and eventually save. And if you don't want, you could trash the annotations that you've added. But this is for users on the command line. So this is just how the search looks like. If you're looking for terms, you'll get the term you searched for, the label that's present in the ontology, description, and then these are the name of the ontology, and this is the actual term. But to make it more user-friendly, we want to put this on the web so that people can then upload their segmentation and then search through a set of terms and then add those annotations to the segment. You could then edit the segmentation. So this is a different example. In order to make it easy to use, we'll have frequently used terms, and then you could add the annotation. And once you've done that, you now have data ready for the volume browser. So let's talk a bit about the volume browser. So the idea behind the volume browser is we want to integrate a lot of data. So the volume slicer is this part here that we saw in the first slide. But now, once we've uploaded the segmentation is available together with a table that shows the annotation and a 3D view of that particular map. And then there's a widget for you to go through the different slices, and you could change orientation. You could view from one side, from the front view, from the side, or from the top. And here we have the segmentations overlaid, and in each of these, we'd have annotation, and I'll show you in the next one of the next slides. So this is the idea. We're yet to add the functionality where we link entries to other entries. So far we have the segmentation table displayed. And if you click on the 3D view, it'll pop out, and you could look at different contour levels, change the opacity, change the color if you want. And it also has a functionality that if you have PDB models associated with this EMDB entry, they will also be displayed together with the map. The table supports interaction, so you can actually click on the table entry and it'll highlight the segment, or you could click on the segment and it'll open the entry on the table. We also have you could set visibility. These are some of the features that we've added. There's still many more that we're going to work on. So there's a lot outstanding. What we need to eventually develop is the processes that people can upload segmentation. So if you need to deposit a segmentation, we need to put that functionality in place. We need to add more file formats that we should convert to EMDB SFF, and I'd be happy to hear from you if you have any file formats you'd like to have translated. We need to put documentation, tutorials, user testing, and we'd like to also convert back from EMDB SFF to various file formats. I think this would be useful because one of the things that we've observed is that through the different workshops we've attended is the different tools that people prefer for different parts of the pipeline. So for example, certain tools are very good for segmentation. Certain types of segmentation, other tools are very good for visualization. And so it would be useful to have a way to convert from EMDB SFF so that people can then take better advantage of the tools that they have. So this is the team, and our thanks to all these people who supported us. Thank you very much.
Great, thanks so much, Paul. Any questions for Paul?
That segmentation file format looks very interesting. But I was just wondering, when you're talking about molecular scale maps, have you
included the option to include symmetry within those. So I'm thinking, for example, a virus structure with manifold symmetry that's been imposed. Can you have just the one segment and the symmetry operators to regenerate that?
Well, presently we haven't factored that in. If that's something you need, because it's an open format, we need as much input as possible, and we'd love to hear what suggestions you have.
Yeah, that would be something that would be really useful.
Excellent.
Very nice talk. Very impressive and futuristic talk, really. It's not on. Okay, so
you can repeat the question
is like, what's the where is the validation and in your software house? Because it's not an exact science to put models in. There's always a qualification of how good the model is and putting different levels together. So how does that appear in your network, where we know the quality of putting higher resolution structures in lower resolution maps? So where's the quality control? How does that read out from your system?
Okay, so your question is about how rendering
is that
visualization at different resolution.
Yeah, so
into the same
that information captured in the quality of that and the validation of that step. How is that in your system?
Okay, so we haven't yet reached the point where we've integrated several data sets together. What we have is we have integrated the maps, contours of the maps, together with PDB models. That's quite straightforward, but what you're referring to is if you have now included different entries into one visualization. So we're yet to think about that problem, but I guess what would end up happening is we'd have to downsample some of the data sets. Where we'll put that information, we're still going to work on that.
Okay, Paul, thank you very much, and we'll catch up with you at coffee time. Okay, the next speaker is Alistair Siebert from eBIC.
Short straw to talk to you all this morning about what we've been doing in the last year on eBIC. So, just a brief introduction before I talk about our previous year's work. So obviously we are your national facility for cryo-electron microscopy, and we were funded by a grant by the Wellcome Trust, MRC and BBSRC, to Helen, Kai, Gert and Dave. We offer free at the point of access to state-of-the-art cryo-electron microscopy via a peer-reviewed process, and you'll see I'll talk later that we've actually revised that a little bit. Predominantly, our mode of operation is beamline-like 24 operation by expert staff, and in doing that enables us to facilitate our user program. We've recently been developing our in-house research under Pei-Jun, and we're trying to play a central role in actually furthering the development of structural biology in the UK with some of the European initiatives, and we're looking to develop further into training programs to really help to assist the community in training, hopefully, the next generation of cryo-electron microscopists. So, last year, I think, or this time last year, we were lauding essentially the efforts of Krios I and its contribution to the community. So yes, we are the people who put a Titan Krios on the experimental hall floor, and that's worked out really very well indeed. So Krios I delivered over 240 days of external use. That's whilst having obviously significant time dedicated for commissioning, for example, the integration of the K2 into the FEI EPU and tomography software packages. So that location has gone from being temporary to being semi-permanent. I confess I always, when asked about it, I'm reluctant to really commit to moving that microscope because it has performed beautifully where it is, and as most of you know, if a microscope is performing well, you really shouldn't move it unless you really have to. So that location is becoming increasingly permanent, and whilst that's the case, we actually took advantage of it to remodel our facility, which at this time last year was still in progress, but now, hopefully, and most of you saw yesterday, is considerably more complete. So actually, we're pretty close to approaching 2 years of, I should read user program, sorry. Krios I was operational for external use in the end of June 2015, so it's almost exactly 2 years now. So I was going to summarise what we've done over the last year. So one of the first things we did was to revise our access model from being entirely rapid access, that being a 48-hour allocation that was peer-reviewed every 3 months, towards moving towards a model that's been very successful in the MX community, that being the block allocation group, whereby a consortia of PIs can apply for guaranteed microscope time and then are responsible for managing their own group of investigators. And the size of those varies from about 5 to 15 PIs. We currently have 7 bags. I think we're getting another 3 or 4 more, and those are obviously occupying an increasing percentage of the Krios time as they do so. We also offer proprietary and industrial access. Currently, that's a little bit less than 5% of our total external user time, and we have the standard diamond cap of 10% on that. Access to the high-end microscopes is obviously based on scientific excellence. We have a superb review panel who are very dedicated and work very hard in reviewing all your applications. And typically about 50% of your applications have been successful traditionally in the rapid route, and block allocation, not sure. So we are very, very fortunate to be funded centrally. So microscope time is obviously free at the point of access for all academic use. And we will actually provide some travel and assistance to UK users, and for European users, if they apply via the iNext pathway, then they can get assistance to travel to us, and then they're free at the point of when they're here. So the first thing we did after CCP-EM, I think it was July 2016, was actually finish up the installation of Krios number 2, which is also in a temporary location on the synchrotron hall floor. First external users that were in July. We have had issues with that microscope. It's approximately 50% of the productivity of Krios I, and there are lots of reasons for that. But one thing we have noticed, a bit of a trend amongst the UK CryoEM community, is it seems that Krioses with even numbers tend to be a little bit flaky. So we'll see how we'll report back on number 4. But yes, so we have been working with the manufacturers to resolve those issues, and we are planning to move that microscope into our beautiful new facility after the installation of the later microscopes. So after the installation of Krios II and the commissioning of that for the user program, we were very excited to see that the building was ready and that we could start installation of our other microscopes. So of course, as mentioned previously, it is a fantastic time to be working in cryo-electron microscopy, and there is huge demand from the community for access to both high-end and screening instrumentation. Our initial remit was to provide, obviously, access to the very state-of-the-art high-end electron microscopes. So to facilitate that, we actually remodeled the facility. As soon as we got the building, we had to remodel it to enable subsequent expansion, but I'll show you a little bit more about that later. So we're very fortunate to have, as I say, a beautiful building, which we share with our friends over in EBIC, who are the physical scientists, physical science electron microscopy facility. They have 2 beautiful JL cold-feg microscopes, and yeah, we're very excited about the potential collaborations there. We also work proximal to I-14, which is the hard X-ray nanoprobe beamline. So it's all part of sort of the diamond strategy for coherent photon and electron imaging. Our facility, So that's the artist's representation of what the building would look like. You see, we're getting closer to it. So obviously that's the main hall with our 4 cryos. And we also have subsidiary labs for other microscopes. We have a lovely loading room that has a very low humidity, where we can also perform plunge freeze and sample preparation. And we have we've started first time this year. We had a ×12 oversubscribed course for cryo sample preparation, and we were kindly supported by FEI with that. So we do actually have, we have access to 8 VitraBots and at Diamond, so we can support hopefully some larger scale training programs going forward, and also working with users to develop projects. And you'll see some of those other microscopes. This one in particular, our lovely little Talos Arctica, is a microscope that we really hope will expand our ability, expand our remit to incorporate a larger community and more users, because a lot of people, everybody's asking, how do we get access to a screening microscope, or how do we develop our projects to the stage whereby they're actually viable for access to the very high-end Titan cryoses. And I think instruments like this would be absolutely crucial. And in itself, it is a very high-performance high-performance microscope. It has an X-Veg emitter, very coherent. It has direct electron detectors. The Falcon 2 will be upgraded imminently to electron counting camera. And we have, I think, just in a fairly routine overnight data collection session on CryoEM, Dan's got a 4-angstrom reconstruction from it. So it really is a very nice microscope in itself. Also capable of some quite nice molecular tomography using the volta-phase plate. So predominantly we've been using that for in-house experiments, for training users, and starting to envisage how our BAG training program will be, our BAG super user training program. And we've had some really nice preliminary industrial projects that have worked quite well on that. And please see our poster on the tour later this afternoon. We also have, we've moved at the start of this year our little Science dual beam, which was housed over in Struby in Oxford, where the optimization of workflows for frozen hydrated lamella preparation were taking place by Kai's group, and we'll be upgrading that system with the FEI loading solution fairly soon. And that should be later in the year. Staff training is ongoing, and a user program is tentatively scheduled for later in the year. I think, well, it says in the poster, I think in late summer, early autumn. And please see the poster for some really nice initial data generated by Korir and Kai's group. As you saw yesterday, we have started to populate our main Krios hall with microscopes. So Krios III installation commenced at the start of this year. We were very fortunate to receive an uplift in our funding from the Wellcome Trust to enable that expansion. So installation at the beginning of this year, and we actually had first users at the very end of February. So that was a really nice installation. Again, that's Alan doing a fantastic job. So it has the same specification as Krios 1 and 2, other than it has an upgraded FEI Falcon 3 detector. And we've started to collect some preliminary data on that, and it's looking quite nice. It will get the K3 upgrade. I'm told by Gatan that will be sometime later in the year, and we're very excited to see that. And please see, so we posted for initial results. It's not initial results from the K3, it's initial results from that microscope. Krios 4 came in just at the start of last month, and that's actually progressed really nicely. The enclosure was built at the beginning of March, and the main microscope was delivered last Wednesday, and they've been making very good progress with that. And that has the same spec as Krios 3. I may have mentioned this on the tour yesterday. I'm very pleased to see that it didn't come in Thermo Fisher colours. We were originally very upset because we thought it was going to be which would have destroyed our beautiful view through the window, because we wanted them all to look the same. So that's very nice. That will also get a K3 upgrade. And whilst we're very fortunate to have lots of microscopes, we're also very fortunate to have had an uplift in our funding to enable more staff to be employed. So Paige was obviously crucial coming to join us at the end of last year. She is the director. We've had Corey, who's specialising in operating the FibSem. Ala will be talking shortly. He's our software guru. And Yuri joined very recently, and Kyle and Jason are here and will be joining us formally at the start of next month. And we also have further vacancies for support scientists and PhD students. So some statistics. You can see it's been very productive. Predominant use is UK-based. We have quite a lot of interest in Europe, and we even have, I think our current record holder is probably from Japan in terms of distance travelled. So we have a strong international use, and that's worked really well. UK predominant use is, as would be expected, from the traditional EM supergroups, and you can see we are currently at, what have we got, almost, over 500 users, over half a petabyte of data, and of the order of 600 days of user time in less than 2 years. So it's been pretty productive. Publications are starting to ramp up, which is really good. There's obviously, we're very fortunate to have fantastic users coming with really nice projects, and there's been some really nice papers that have been published from that. And we're now at, I think, 10 plus 2 more facility papers. So that's really coming on very, very nicely. So please keep us updated if you do publish something, and we're very excited to see all the work that's coming out. So just to conclude, these are the people who've played the predominant role in obviously bringing eBIC to the state where it's at now. Also, I would like to thank, we are very fortunate to be on a very large-scale facility, so we have substantial support from all of the Diamond team, and the computing unit have been very, very helpful as well. And obviously, for microscope installations, Alan has spearheaded the Krios installations, and Alberto working on the Talos and the SIAS. And we've had a lot of support from FEI in terms of application support as well, so that's been brilliant. And of course, all of this has been directed by Pei Jun, and with the eBIC management team of Dave, Kai, Helen, and Martyn. We have also, that's Yuri and Alla, and that's Kyle and Jason joining shortly. So I should probably conclude and pass over to Alla, because he's going to talk about data processing. Should we do questions now, or questions at the end? What do you prefer?
We can take a quick question now.
Quick questions now?
Any questions for Alistair now?
So you have a wonderful new building for your Krioses, of which you have space for 4. Surely that's not enough for a national facility. What next?
What next? As I said, Krios I has performed beautifully, and its temporary location is looking increasingly permanent. So we'll see when that gets moved into the facility. If that doesn't move into the facility, there will be obviously space for another one there. And then Krios II is also a subject of some debate. So there is potential room for expansion as needed. Obviously, we are fortunate to be on a site that does have quite a lot of empty space around it. So I'm sure we're very excited to, obviously, the Rosalind Franklin Institute is another very exciting development that will be on site fairly soon. So I think there's plenty of room to build, and I always think Electron microscopes are a fantastic bargain when compared to an XFEL. I probably shouldn't say that, sorry. Microscopes for micro-ED. It would be lovely to have a dedicated microscope for micro-ED, yes. The instruments that we have currently are very flexible and very powerful, and are capable of micro-ED, and there are some nice preliminary data presented on, I think, Korir's SIOS poster showing some micro-ED. So yes, we'd love to have a dedicated micro-ED microscope. That's a fantastic idea, but we are looking to expand, obviously, work into that area, and I think that's something that, yes, the industrial unit will also play a large role in. So, yeah, that's a very exciting area for EM.
Okay.
Thank you.
Thanks, Alistair. Good to know that there's more room around the periphery of the ring. And without further ado, we will introduce the software guru, Al-Afee.
Thank you very much. Hello, everyone. My name is Ala. I'm a data analysis scientist in eBIC. Today, I'm going to give you an update about the data analysis infrastructure in eBIC. So my agenda is going to be, I'm going to talk about the infrastructure, a general view of the facilities that we have, the computing and the softwares. We have two options for doing the data analysis. We have a manual option and we have an automated processing pipeline. And finally, I'm going to show you some examples about the data that have been generated and the challenges that we have at the moment. So this is the network diagram of the microscopes. So you can see that we have a microscope that has a dedicated machine on it, which is connected to a very high-speed network. Now, this network is connected to our GBFS file system, which is a very high-speed and parallel file system. It is connected via an InfiniBand low-latency network, which is very high speed, especially when it's connected to our clusters. And, of course, you can access all of this facility from an outside network environment in a secure way. This is a more detailed diagram. So you can see that the microscope has 3 dedicated machines. So we have a machine that is dedicated for the detector, we have a control computer for the microscope, and we have a rack-mounted server for the automated processing. Now, once the data come from the detector, it will be stored locally on the local workstation, which will be transformed to our GPFS system via an rsync script in a real-time fashion. Also, our storage system has a connection with the central computing. Again, it's an InfiniBand low-latency network. Our central computing consists of around 2,500 CPU cores. Each core has up to 12 gigabits of RAM attached to it. And, of course, we have a 86 GPU. So eBIC has an access to this, and also eBIC has a dedicated central computing facility, which was installed in the last shutdown. Yeah, so now I'm going to talk about the data processing options. Once our user come to us and start collecting data, it is possible to do manual processing. Okay, so we have a variety of softwares have been installed. Thanks for the CCP-EM for helping us on this. We have a modular system, so you don't need to install any software. Just you need to type module, load, and then the name of the package, and you'll be able to use this software. Of course, the software can access the data on the GPFS system, also have access to the cluster. So many of these softwares has been configured, so you don't need to bother about which node on the cluster to use. Or, of course, you can submit job to the cluster node. You just need to module load the global cluster and then a qsub command. Of course, you can access the data either from inside Diamond or from outside via no machine within a specific period of time of your visit. The second option is the automated pipeline. And following the great success of our NX beamline automated processing, Diamond started to be interested in auto-processing of EM data from eBIC. So basically the idea here is that once the data collection is started, this data will be automatically processed through the pipeline so that you can see the result, or at least some result that guides you through the acquisition process. Now, once the files or the images or the frames, let's say, is available on the GPFS system and some metadata on our I-SPY-B database, the process or the processing of these data will be automatically picked up by our software, and then we will, of course, this software will send messages to our queue. Now, why do we have a queuing system? Because our central processing is assured resource. So all the beamlines in the synchrotron use the central computing facility. So we need to use a queuing system so that we can efficiently manage this computing facility. Now, basically, usually in eBIC, there's two kinds of experiments happening. So there's the single particle and there is the tomography experiment. So based on the file, the pipeline will be able to determine what kind of experiment it's happening and then send it to the corresponding pipeline. Now, of course, software development is not easy in such an environment. It's extremely hard. So we need some tool at least to help us to at least find out what is the problem and try to tackle it in a timely fashion. So one of these tools is the logging. So we have something called the gray log. So what we do is that whenever we write something, we just write logs into this database and then have a look on it to try to understand what's happening in case if there's a problem. Also, we have a testing system called JSON. So basically, our software is developed by multiple developers. Definitely, these developers are human and can do mistakes. Now, once there's a mistake, we need to at least find as soon as possible that there's a mistake. So this kind of infrastructure will help us to spot these mistakes immediately and try to solve it as soon as possible. Now, more details about the single particle pipeline. So the single particle pipeline start with, of course, the creating the stacks from the frames first, and then, of course, start the beam-induced motion correction. Now, currently we are relying on Motion Core 2. Following this, the CTF estimation to find out the defocus value and, of course, find out if there's an astigmatism in the image and tell you information about the quality of your tomograms. Now, these three steps is implemented now and it's live. However, we are currently working on the particle picking. Particle picking, unfortunately, it turns out it's not an easy step, and especially with our kind of requirement. So, when you're talking about automated pipeline, you need the lowest amount of input from the user. And of course, we don't want any kind of manual intervention from the user. Otherwise, it's not going to be automated pipeline. So we are having an issue with this. But once this step is being implemented, it's going to be straightforward to go to the next steps, which is 2D and 3D classification, and of course, 3D refinement. I should mention here that so the initial view of the pipeline was just to give you some guidelines about your data. Is it useful or not, and try to guide you through the acquisition. However, over time, the result from the pipeline becoming more and more important, and many of our users is interested to take the data from the pipeline. So we have implemented a user template. So it's possible for you to change the parameters for the reconstruction or the pipeline, so that you can, for example, if you are interested to take the motion core data, it is possible for you to edit the dose information so that you can correct for it and then start your manual processing when you go back to your home institution from there. That is a possibility. This is an example of the software that we use. So we have our own softwares, but again, we don't want to reinvent the wheel. So one of the nice softwares out there is Symposium. Symposium is a software that can integrate different EM softwares. And of course, we created our own version of Symposium. We call it the headless, and it's available open access on the GitHub. It's possible for you to download it, and feel free to contribute for that. Of course, all the processing is being stored from each step is being stored on our GPFS system. So it's not really dependent on each other. The result right immediately. Yeah. And then the second pipeline, which is the tomography pipeline. So tomography pipeline start from the motion corrected. So once we motion correct the images and then we generate a stack, we will immediately send it to iMode. Of course, iMode is the standard software for tomography in EM. And there's new releases of iMode that support auto-processing and batch processing, which is very helpful in our case. And then after all these processing is being done, it will be stored in our iSpyB. Now, our iSpyB is a database that has been developed in Diamond and actually used by all the beamlines in Diamond. And basically, it stores the metadata and the results from the experiment. So from the user side, this is an example of tomography experiment. From the user point of view, you'll be looking at your data while acquisition. At the same time, you can log into iSpyB via web interface or even an iOS app on your iPad to look on what's happening on your data. So all kind of processing is available for easy access. Now I'm going to talk about the data issues, or the amount of data that is being generated in eBIC. So this is an example from just one microscope we call the MO2 or Krios II. So you can see for about 1.5 years now, we are almost approaching a 1 petabyte of data. You can see the majority of these data is coming from the raw images. Of course, our users do some manual processing, and the automated processing just started recently, and it's also contributing to this curve. So you can see we are talking about 1 petabyte of information in about 1.5 years. Now, how does that compare to Diamond? Now, in Diamond, we have about 7 petabytes of data generated by all the beamlines, and just one microscope is about reaching the 1 petabyte barrier. Now, of course, that is a huge amount of data from one beamline. And of course, that is a good thing because it's going to generate scientific advances. But of course, with such big blessings, we will have big problems in tackling these volumes of data. This is an example of what if we are going to upgrade one of our detectors. So here we have this microscope have a K2 Gatan detector. There's plans to upgrade it into a K3. So K3 is theoretically going to provide us with 4 times the size of the data. You can see the theoretical jump is going to be around 3.7 petabyte of data, which is huge. And then we have other microscopes, and then it's going to be very challenging to tackle it. So we are working on tackling these challenges at the moment. And finally, I'd like to thank the groups that I'm working with. So I'd like to thank the head of the data, the software science data group, Diamond. Also, I'd like to thank eBIC group. And of course, the developer of CCP-EM for their help. And of course, Struby Oxford. And finally, CCP-EM for their help. And yeah, that's pretty much everything.
Great, Al. Thank you. Okay, I think we can save a number of questions to coffee, except if somebody has a burning one they want to be heard around the world. Okay, so we'll continue the conversation in the atrium over coffee. I'd like to thank all the speakers this morning for their excellent presentations and also the fantastic work behind that. That's really helping all of us. Thanks a lot.